Thanks, everyone. My name is Nancy Jones. I'm the Vice President of Clinical Development at Neuren Pharmaceuticals, and it's my pleasure to be able to join you today via this recorded presentation to give you an update on our clinical development program for Angelman syndrome. Neuren is a specialty pharmaceutical company, and we are specifically focused on the development of treatments for neurodevelopmental disorders. We have two compounds in development, trofinetide, which you see here at the top, and NNZ-2591. Trofinetide actually has been approved for the treatment of Rett syndrome in the United States, and that is with our partner, Acadia Pharmaceuticals in North America, and it is available in the United States as Dabu. NNZ-2591 is in phase 2 development for neurodevelopmental disorders, and it is NNZ-2591 that's being developed for Angelman syndrome. So where does NNZ-2591 fit in with other medications that are being developed for Angelman syndrome? When you're developing new medications, you can either repurpose existing drugs or have new drugs. And these can be drugs that are developed specifically directly at the genetic dysfunction. You can have neurobiological therapies, which are focused on treating the effects on the brain, the neurological effects, and you can have treatments that are for specific symptoms. And NNZ-2591 is a novel neurobiological therapy. So specifically, NNZ-2591 is a novel synthetic analog of cyclic glycine proline, which is a metabolite of insulin-like growth factor 1, or IGF-1. So IGF-1 is a growth hormone that occurs in your body and is present in the brain, and it is responsible for managing the growth and development of the cells in the brain. And when it occurs naturally in the body, it breaks up into smaller pieces, which are called metabolites. And one of these metabolites is cyclic glycine proline, or CGP. And what NNZ-2591 is a synthetic analog, which means that it has exactly the same molecular structure, with the exception of one small change. And that one small change gives it properties that make it better to be used as a medication. So what this means is that it can have the same actions as a naturally occurring molecule. So in this case, it's able, like a naturally occurring molecule, to regulate the amount of IGF-1 that's available to activate IGF-1 receptors. But it has the properties that you see on the right that make it good to use as a medication. So it's orally available, it can be taken orally. It is actually a liquid formulation. It has 100% oral bioavailability, and that means that all the drug is able to be used and absorbed. It crosses the blood-brain barrier, and it has good pharmacokinetic properties. And that essentially just means how the drug is used and absorbed by the body. So where are we at in terms of our progress in terms of clinical development program? So what you can see here in green are the steps that we have completed, and the one in purple is ongoing. So from our preclinical work, we have been able to show what NNZ-2591 does at the molecular level. And what we found is that it improves the abnormal function of brain cells. So specifically, these cells called microglia cells, which support the neurons. It improves the function of those cells, and it rescues abnormal morphology and density of dendritic spines. And the dendritic spines are the parts of the neurons that actually receive the messages from other cells. And when either the shape of them or the density of them is abnormal, if you treat with NNZ-2591, it normalizes them. This actually then shows and we were able to show that then there's an improvement between signaling between cells. So if it's impaired, the signaling, it gets improved by treatment with 2591, and then 2591 also improves bioavailability, or the availability of IGF-1. So from an animal model study, which was a mouse model of Angelman syndrome, we found that treatment was able to fully rescue the phenotype in this mouse model. And that included seizures, as well as the behaviors that you see in the graphs here, which are all representative of analogous behaviors that are similar to symptoms that you see in individuals with Angelman. So you've got hyperactivity and anxiety, you've got measures of sort of daily living or functional capabilities, sociability, motor skills, and cognition. All of these things were sort of normalized in treatment with NNZ-2591 in the mouse model of Angelman syndrome. So for our clinical studies, we have completed the phase 1 study, which is the first study you do to look at safety and also sort of targeting the dose. And this study was in healthy adults. There was a single dose stage and then multiple ascending dose stage, which was looking at both a higher and a lower dose. And it was 7 days of twice daily dosing of either NNZ-2591 or a placebo. The safety results from this study were good. Adverse events were all mild or moderate and resolved during the trial. There were no serious adverse events or severe adverse events, and no clinically significant findings on lab tests, vital signs, or cardiac tests. The most common adverse event was drowsiness, and there was one discontinuation which resolved after the treatment was completed. The PK results are what we want to see. Is essentially we wanted to see a predictable PK profile. So currently we are at phase 2, and we have enrollment open in our phase 2 study with top-line results expected in next year. So the phase 2 study is 13 weeks of treatment, and it's an open label study, which means everyone is getting NNZ-2591. It's oral dosing, it's a liquid formulation, and then the treatment is taken twice a day. And there are in-home nursing visits as well as in-clinic visits as part of the study. So the primary aim is to evaluate safety and then also looking at pharmacokinetics, which, as you may recall, is how the drug is absorbed and distributed throughout the body. But we're also looking at the clinical response to the drug, or sort of the signs of efficacy. And we're looking at a range of outcomes that are all important symptom areas in Angelman: communication, fine and gross motor skills, behavioral issues, GI, sleep, seizure, and then measures of a daily function. And the findings we have from this study will inform the design of pivotal trials, or those larger trials that would follow if the phase 2 study is successful. So here you can see a snapshot of some of the key inclusion and exclusion criteria. If you want to see more details, there's information is posted in detail on clinicaltrials.gov. So this trial is looking at ages 3 to 17 in both males and females, with individuals that have a diagnosis of AS with a confirmed genetic etiology, a minimum weight at screening, and of course, the ability to swallow the liquid study medication, and then a stable seizure profile and treatment regimen. At screening, the participants also need to do assessments. There's a review of current medications, as well as a review of the medical history, which is also used to determine the eligibility for the study. So the study sites are all in Australia. So we have 3 sites, one in Brisbane, one in Melbourne, and one in Sydney. And there is a study-specific website, angelmanstudy.com.au is also where you can find helpful information if you want to learn more about the study. And then, as I mentioned before, at clinicaltrials.gov, you can get to our study by searching Neuren and Angelman. Also, if you're interested in updates about the study, you can sign up on the Neuren website, neurenpharma.com, for alerts. And this is found under news and reports, and then you can click on email alerts. And then if there are any updates posted, you'll receive an email with that update. So thank you very much for your attention, and enjoy the rest of the conference.